New research has surfaced regarding hair loss in men. The study has revealed that rare genetic variants play a role in male pattern hair loss. Male pattern hair loss, colloquially known as androgenetic alopecia, progresses from a receding hairline to baldness on the crown, followed by the classic horseshoe-shaped hair loss pattern. Previous studies have established multiple commonly occurring genetic variants that contribute to this condition. Human geneticists from the University Hospital of Bonn and the Transdisciplinary Research Unit Life and AMP Health of the University of Bonn conducted a systematic investigation to determine the impact of rare genetic variants on this disorder. The study examined the genetic sequences of 72,469 male participants from the UK Biobank project and identified 5 genes that had a significant association with the condition. Additionally, the study confirmed the genes that were already implicated in earlier research. The results of the study have been published in the scientific journal Nature Communications. Male pattern hair loss is the most frequently occurring form of hair loss in men and is mainly attributed to genetic factors. Unfortunately, the current treatment options and risk prediction methods for this condition are not optimal, and therefore further research into the genetic basis of this condition is essential. To date, most studies have focused on common genetic variants and have identified over 350 genetic loci that may contribute to this condition, with a particular emphasis on the androgen receptor gene located on the X chromosome inherited from the mother. In contrast, the role of rare genetic variants in causing this condition has been assumed to be minimal, although this assumption has not been systematically investigated. Such analysis presents a greater challenge as it requires a large sample size, and the genetic sequence must be captured base by base using genome or exome sequencing of affected individuals, explained Sabrina Henner, a doctoral student at the Institute of Human Genetics at the UKB and the University of Bonn. The statistical complexity arises from the fact that rare genetic variants can be carried by very few or even a single individual. The methodology behind gene-based analyses involves the consolidation of variants based on the gene in which they are situated, as elucidated by corresponding author PD Dr. Stephanie Heilmann-Heimbach, who is a research group leader at the Institute of Human Genetics at the UKB at the University of Bonn. Bonn researchers utilized various methods, such as the Sequence Kernel Association Test, SCAT, a prevalent method employed to discover associations with rare variants, and GenRisk, a technique developed at the Institute of Genomic Statistics and Bioinformatics, IGSB, at the UKB and the University of Bonn. The study involved the analysis of genetic sequences from 72,469 male UK Biobank participants. Within this comprehensive data set, geneticists from Bonn, in collaboration with researchers from the IGSB and the Center for Human Genetics at the University Hospital Marburg, scrutinized rare gene variants that occur in less than 1% of the population. Using modern bioinformatic and statistical methods, they discovered connections between male pattern hair loss and rare genetic variants in 5 genes: EDA2R, WNT10A-HF, SEPT1, and EIF3F. Before conducting the analyses, EDA2R and WNT10A were already identified as candidate genes based on prior research on common variants. Dr. Stephanie Heilmann-Heimbach explains that our study presents further evidence supporting the assertion that these 2 genes play a role in male pattern hair loss and that this role is fulfilled by both common and rare variants. Similarly, GPH is situated in a genetic region that has been previously linked to male pattern hair loss through common variants, specifically the EDA2R/androgen receptor region, which has been consistently associated with this condition in past research. Sabrina Henner notes that, although HEPH has not been previously considered as a candidate gene, our study suggests that it may also contribute to male pattern hair loss. On the other hand, the genes SEPT1 and EIF3F are positioned in genetic regions that have not been associated with this condition before. Therefore, they represent entirely novel candidate genes, and we posit that rare variants located within these genes may contribute to the genetic predisposition for male pattern hair loss. Given their known role in hair growth and development, HPH, KEPT1, and EIF3F are highly plausible new candidates for this condition. In addition, the findings from the research imply that genes responsible for causing uncommon inherited disorders that affect both hair and skin, such as ectodermal dysplasias, could potentially contribute to the onset of male pattern hair loss. The scientists involved in the study aspire that the fragments of the puzzle they have uncovered will aid in the comprehension of the underlying reasons for hair loss, and in turn enable more dependable risk assessment and enhanced treatment methods. Funding for the research was provided by the medical faculty at the University of Bonn. Prof. Dr. Marcus Nothan, the co-author of the study and director of the Institute of Human Genetics at UKB, is a member of the Transdisciplinary Research Area, TRA, Life and Health, at the University of Bonn. The expenses for the publication to be available in open access format were supported by the DEAL initiative of the University of Bonn. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.